Hello, and welcome to part one of the lecture on chapter 4, which is cell physiology and pathophysiology. Share my screen and I'll call up the PowerPoint. All right, so hopefully you can see that first slide. So this topic is multiple topics in one. So we're going to break it up into 4 main topics. I will cover the first one in part 1, and then I think we're going to do 2, 3, and 4 in part 2 video. There may be a part 3 if it gets too long, but I'll try to keep these brief so that you can do them when you have a little bit of time. So the first topic is cell transport, that is moving things through the cell membrane. Then we'll get into cell signaling, what molecules are used to send messages from one cell to another and how those messages are received. Then this topic is a little bit weird. We're going to talk about muscle cells and how they work. It fits into this chapter because we're talking about cells, but it's a little bit of an odd man out. And then lastly, we'll talk about stressors that happen to cells and how cells can adapt to stressors, how they get damaged by stressors, and how cells die. All right, so start with cell transport first. This kind of buff colored background, I'm going to use that at the beginning of each of the topics so you have an idea that we're changing topics. So this first one is cell transport. Some general biology, some basic biology. Every living thing is made of cells. There's a phospholipid bilayer that is phosphate heads with lipid tails. Those two structures face in towards each other. Those lipid tails face into each other. That's the hydrophobic barrier, those lipid tails of the phospholipids. And they prevent water from inside the cell and outside of the cell from mixing with each other, as well as the things that are dissolved in that water. Now, if something is small and hydrophobic, for example, oxygen, carbon dioxide, both of those are very small molecules and they don't have a charge. They share electrons equally. There's no charge. That stuff just passes through the membrane as if the membrane weren't there. Steroid hormones, same thing. These are lipid-based, and so they can pass right through that membrane. Everything else is going to require a specific transport protein, a membrane transport mechanism. When items are moving against the concentration gradient, that means from an area where there's not a lot of them to an area where there's already a lot of them, you have to push that. That's going to require an embedded membrane protein, but you're also going to have to pay for pushing something against its concentration gradient. So a source of energy, that source of energy is usually ATP. Then if the molecule, this hydrophilic molecule, is going to move down its concentration gradient, I don't have to pay for it every time to get it down the concentration gradient. However, because it's hydrophilic or polar, it's not going to be able to diffuse right through the membrane with no problem. So I need what's called facilitated diffusion. I need a specific protein that's embedded in the membrane, but I don't have to provide energy every single time. There's a general diagram of the cell. The things that are important for our purposes today are this phospholipid bilayer, this outside part. And we're going to talk a little bit about some of the organelles, the mitochondria. This is where a lot of the ATP gets made. We'll talk about the ribosomes that are used for translation and the fact that they're stuck on the outside of this endoplasmic reticulum. There's areas where the ribosomes are stuck. That's the rough endoplasmic reticulum, or rough ER. And then areas where we don't have ribosomes, that's the smooth ER. And especially when we talk about muscle cells, we'll talk about the role that this organelle has. I think those are the most important structures we'll deal with today. So a reminder, what we mean by a phospholipid bilayer, we've got phospholipids, these blue balls represent the phosphate heads and the little tails poking down towards each other. This is the inside of the membrane. That's the lipid core. That's the part that's a real barrier between the outside of the cell and the inside of the cell. There are embedded cholesterol molecules. Those are the things with like 4 ring structures. These are sterols. That's the sterol part. Steroid hormones are based off of cholesterol. You make them from a cholesterol base. And those things are lipid soluble. And then we've got proteins that are embedded here in the membrane. They have actual jobs. For example, this one looks like a channel protein. It's going to let something through the membrane for facilitated transport. Those are the important things to know. Lipid center, embedded proteins that do particular jobs. The organelles, as I mentioned, there's a nucleus. That's where the DNA is. That's where DNA gets copied. So DNA replication. That's where DNA is read to make a message that can leave the nucleus. That's the process of transcription. Some other organelles that we'll talk about are the ribosomes, as we mentioned. They're stuck on the outside. Some of them are stuck on the outside of the endoplasmic reticulum, forming the rough ER. We have smooth ER that plays a major role in calcium storage. Look for this when we get to muscle cells. The mitochondria, the structures that make the ATP, and the lysosomes. These, I think of them as like little cellular stomachs. They've got degradative enzymes, things that can break down structures that we want to get rid of. And this becomes an important part of what happens in cell damage and why sometimes cells cannot recover from damage. Okay, this information you can find on pages 76 and 77 from our textbook. All right, so moving things through the membrane. When we move hydrophobic molecules, as I've mentioned, examples, oxygen, carbon dioxide, lipids, these molecules move by diffusion. They just pass right through that membrane. They travel down their concentration gradient. Everything else is not going to be able to diffuse directly by simple diffusion. It just can't get through that membrane. And so let's talk about what happens with the different hydrophilic molecules. The things to consider are the size of the molecule and the charge of the molecule and the concentrations of the molecule on the 2 sides of the membrane. So for something that's a large, like for example, proteins, they have to move by a process of either exocytosis or endocytosis. The difference is what direction is it moving. If it's made inside the cell, the protein's made inside the cell and it's being exported, we call that exocytosis. If it's something that's outside of the cell and we want to bring it in, for example, a white blood cell engulfing a bacterium, we call that endocytosis. If the molecule is smaller than that, something like, for example, glucose or amino acids that we are going to be needing to bring into the cell so that we can make proteins, these can pass through if we make a channel for them, if we make a transport protein. That process of passing through a channel like that, that's what we call facilitated diffusion. If the thing is moving down its concentration gradient. If we have a protein embedded and we're moving it through the membrane, but we're moving it against the concentration gradient. If we already have a lot of that stuff on one side of the membrane and we want more on that side of the membrane, that's going to have to be pumped. That's going to be through a process known as active transport, where we have to pay that protein with an ATP every time to get something through. All right, if what we're talking about is tiny, but has a charge, like ions, for example, also known as electrolytes. We're talking about sodium, potassium, calcium, magnesium. Okay, those are the most common ones. They're involved in ion channels. And remember, ion channels means facilitated diffusion. It's still moving down its concentration gradient. That's where the diffusion part comes in. And the facilitated is the fact that we had to build a channel for it. We had to have an embedded protein that's in the membrane. Okay, so if it's going down the concentration gradient, it's facilitated diffusion. If we're going against the concentration gradient, that's when we call it active transport, because we're going to have to pay every time. And examples that we're going to see are like the sodium potassium pump. That's an active transport mechanism for ions. Now note, water, typically, although movement of water is very important in the human body and shifts of water, shifts of fluid are very important. Interestingly, water typically moves through the process of facilitated diffusion. So we build a channel for it, and that channel, as you'll see, is called an aquaporin. But we don't usually pump water. What we do is move ions around to encourage the water to move. So water will move from an area of lower osmotic strength to areas of higher osmotic strength. Another way to think about that is water follows sodium. So wherever you have a lot of sodium, water tends to be drawn in that direction. And so to move water around, sometimes what we'll do is pump sodium and then let the water follow that. Okay, this information, pages 77 to 84 in the textbook. This is just a diagram showing diffusion through a membrane. So if there's a lot of this molecule on the outside, these are all oxygen molecules, they can pass freely through that phospholipid bilayer because they don't have a charge. They're also hydrophobic, so they pass right through with no problem. So that's just simple diffusion. Then we'll march through the different processes. We said endo and exocytosis. This is the movement of large molecules like protein. Endocytosis, you'll recall, this is inside the cell here in blue. So endocytosis is the process of the cell capturing something large and bringing it into the cell, the arrows indicating bringing it in. The next two are both examples of exocytosis. Again, the blue is inside the cell. This is something that was made inside the cell and then we want to ship it out. Let's say we're talking about liver cells making albumin, for example, or clotting factors, or many, whatever of the many tens of different kinds of proteins that liver cells make. And they're releasing them outside of the cell membrane. That's through the process of exocytosis. It's just going on continuously. In contrast, we can also do regulated exocytosis. It's a similar process where we make a protein, for example, some kind of hormone, insulin, for example, it's a signal molecule that we're going to send out. We don't send it out just willy-nilly. We tend to send it out when environmental conditions are such that we need to release some insulin. And so here in yellow is representing inside the cell. And so a protein's been made, it's been packaged up in this vesicle. Once the cell has the appropriate conditions, that vesicle moves towards the cell membrane, fuses with the cell membrane, and eventually releases the contents of that vesicle. And this is what we do with some hormones as well as neurotransmitters. So this is happening at the ends of axons. There's vesicles that have the neurotransmitter already made, and it's just waiting to receive the nerve signal, and then that can be released. And again, that's exocytosis. Facilitated diffusion. So what it has in common with exocytosis or endocytosis is we're moving something through the membrane. In this case, though, the thing is small enough that we can get away with what I think of as a revolving door. It's just something that kind of opens and closes and lets the material through without leaving a giant gaping hole in the cell membrane. These structures are pretty specific for whatever material they're transporting. One example, an easy one, a common mechanism of glucose entry into cells. So we mentioned insulin is that signal. The things that cells do in response to insulin is they put these transport proteins into the membrane, and that's what allows the glucose to come into the cell down its concentration gradient. There's a lot of sugar outside the cell and not so much inside, and so the glucose goes down its concentration gradient, but it needs a tunnel, it needs a revolving door, it needs a way to get in. And then active transport. The example we're using here is the sodium potassium pump. You've heard about it a million times in your biology classes. It operates continuously, or they operate continuously. So their proteins are embedded in the phospholipid bilayer. We have to pay for this movement every time. So we hydrolyze ATP. That means we take remember, ATP stands for adenosine triphosphate. It's got 3 phosphate groups, and we hydrolyze, meaning we take off one of the phosphate groups. Now we're down to ADP, adenosine diphosphate. So that process of hydrolyzing the ATP energizes, if you will, this molecule and allows it to change shape. So it's got these 3 sodium ions on it. The changing of the shape pumps those 3 sodium ions out. Before that thing can take 3 more sodium ions out, what it does is it brings a couple of potassium ions, these green triangles, or not triangles, what are they? Diamonds, brings the green diamonds into the cell. And then again, it's ready to pump 3 more sodium ions out. This thing's going all the time to maintain that difference in sodium and potassium charges outside and inside the cell. And we pay for it all the time. All right, another couple of active transport examples that I'm going to mention in passing. There's a pump for calcium ions, and there's a pump for hydrogen ions. And the reason I'm mentioning both of those is there are actually medications that block both of these pumps, and so I just thought I'd mention them. All right, so we've got calcium pumps. We have calcium pumps in 2 main places. One is along the plasma membrane, and we're going to talk about these when we talk about the calcium. Well, you've heard of calcium channel blockers, those medications. Those are the calcium channels that those medications block. It's the calcium channels that are on the cell membrane itself. There are also calcium channels on the endoplasmic reticulum. We can talk about that when we talk about cells. And the other one that I wanted to mention is this proton pump. You've heard of protons. Oh, reminder what protons are. Remember, when we talk about these things, we're talking about ions, which means it's an atom, but the number of protons and the number of electrons don't quite balance each other. So there's a charge on it, and that's what makes it impossible for this thing to just spontaneously pass through the membrane. That's why it needs a channel. Hydrogen, an atom of hydrogen is 1 proton and 1 electron. So if you change the electron, you just have 1 proton. So sometimes you'll see this called hydrogen ions, and sometimes you'll see it called protons. It's the same thing. All right, so hydrogen ions being pumped from one side of the membrane to the other. This happens, for example, in the stomach to produce stomach acid. And there are medications, PPIs, proton pump inhibitors that interfere with the pumping of those hydrogen ions across the membrane. Amazing. Both of these pumps, they're for active transport, so they both use ATP for the energy to transport the molecules or ions. And I think this is the last point in this section, and that is the water molecules. Remember, water molecules are an oxygen atom represented by this red circle and the 2 Mickey Mouse ears. Those are hydrogen atoms. So it's H2O. This thing is polar. It is not going to pass easily through the cell membrane because of the lipid barrier. However, if we build this channel known as an aquaporin, which literally means water hole, the water molecules can pass quite easily through the cell membrane. So polar water molecules diffuse freely across the cell. So for example, another way of thinking about this is more dynamic than this picture indicates. Whether the aquaporins are present or not, or how many are present, varies. The cells can manipulate that depending on which way they want water to move. So for example, regulating how many aquaporin proteins are present contributes to how the kidney regulates how much water is being released in the urine. So in reaction to a hormone, vasopressin, hormone also known as ADH, and antidiuretic hormone. So antidiuretic, you want to withhold more of the water, don't pee out all the water. Vasopressin is a hormone, it's also a drug, and it's used to keep more of the water in. So basically the cells of the kidney tubules are told, hey, put a bunch of water channels in here because we need to recoup some of that water that otherwise is going to go out in the urine. So putting in more aquaporins allows the water to be retained. Oh, so one more slide before we take a little break. So the channels. So we build these proteins and embed them here in the phospholipid bilayer. And that allows ions or charged particles, these ions can move down their concentration gradient through the channels. This is another example of facilitated diffusion, like the aquaporins. They act like pores. When they're open, ions can freely diffuse downhill, if you will. So from areas where there's a lot of them, for example, out here, to an area where there's less of them in here. The channels are pretty specific for a particular ion. They'll let sodium through, or they'll let potassium through, but they're not just general, anything can get through. And how this is different from what's there for glucose is these are open, can stay open continuously. An easier way to think about this is the thing that you need for glucose or amino acids. Those are pretty big structures, multiple atoms bonded together. So I think of that as a door, and I don't want to leave a door all the way open because then all kinds of stuff can get through. So that's where we go with the revolving door, the thing that can open and close, but it's not just a giant gaping hole. In contrast, when we're talking about ions, we're talking about one atom. And so that opening is much smaller. So it's like a small window that you can leave open, but only certain things can get through, or maybe think of it as a doggy door that's specific for this kind of doggy that can get through. And there we will wrap up the cell transport. This will take us to slide 15, and we'll pick up here for part 2 of this recording.